Assalamu alaikum. Bismillahirrahmanirrahim. I'm Dr. Kuratullah and I'm pharmacist by my profession. And this presentation is prepared by me. I'm Form D, Research and Development Manager, and doing my regulatory affairs in RAP. I also do consultancy online and a business unit head. Today of our topic of discussion is receptors. What are receptors and what are the functionings and what are the different strategies and types of receptors? First of all, a receptor is a macromolecule in the membrane or inside the cell that specifically chemically bind a ligand or a drug. The binding of a drug to receptor depends on types of chemicals bonds that can be established between drug and receptors. Main action ways of receptor. What are the main action ways of receptor? These are following. There are the three main ways the action of the receptors can be classified: relay of signal, amplification, or integration. Relaying sends the signal onward. Amplification increases the effect of a single ligand, and integration allows the signal to be incorporated into another biochemical pathway. These are basically the action ways of a receptor. Now next, there are certain locations of receptor. What are the specific sites where receptors are located? These are receptors proteins can be classified by their location. Transmembrane receptors include ligand-gated ion channels, G protein coupled receptors, or enzyme-linked hormone receptors. In the continuation of location of receptor, there are also intercellular receptors. These are found inside the cell and include cytoplasmic receptors, nuclear receptors. This is the pictorial diagram that you can see. There is a two synaptic clefts, postsynaptic receptors, and the uh, mm, presynaptic receptors. In the diagram, you can see there are calcium vaulted gated channels, and there are also certain receptors uptake pump. Synaptic cleft are also there, and this is the postsynaptic receptors. Neurotransmitters are released by presynaptic receptor, and these are the site which are located postsynaptic receptor. These are the site which basically interact with the neurotransmitter and perform their action. What is the ligand? Ligand, a molecule that binds to a receptor, is called a ligand. Any type of molecule that bind to the receptor, this is called ligand, and can be protein, peptide, or short protein, or other small molecule such as a neurotransmitter, hormone, pharmaceutical drug, toxin, calcium ion, or parts of the outside of virus or microbes. These all categorized into the ligand. There are also major categories of receptors. Type 1, ligand-gated ion channels. What are the ligand-gated ion channels and how they perform their function? We can see in this slide. These receptors are typically the targets of fast neurotransmitters, such as acetylcholine or nicotine and GABA. Activation of these receptors results in changes in ion movement across a membrane. Type 2, these are basically G protein coupled receptor. This is the largest family of receptors and includes the receptors for several hormones and slow transmitters, for example, dopamine, metabotorographic glutamate. This is the pictorial diagram again. You can see there are this in the picture dopamine receptors that when receptors receive certain signals, then they can release dopamine. In the pictures, dopamine metabolites are also seen. Dopamine, then synaptic cleft is shown in the figure. Dopamine then received by the synaptic vesicles to produce their certain actions. Type 3 kinase ligand and related receptors. They are composed of an extracellular domain containing the ligand binding site and an intracellular domain, often with enzymatic function, linked by single transmembrane alpha helix. The insulin receptor is an example. Insulin is basically perform their function via kinase-linked and related receptors. In the picture, you can see the function of insulin receptors. There are also certain beta sites and alpha sites. insulin basically accepted by the alpha receptors. After insulin attachment with to the alpha sites, then signaling pathway activates its function. There are also, you can see, glucose is attached via this receptor site. These are basically glucose transporter glutamine. When insulin bind to the receptor, these sites blocked ultimately, and insulin releases or produces their action. Type 4 nuclear receptors. While they are called the nuclear receptors, they are actually located in the cytoplasm and migrate to the nucleus after binding with their ligands. Steroid and thyroid hormone receptors are examples of such receptors. Basically, our thyroid hormone, thyroid stimulating hormone TSH, and many steroids that are basically produced in our body, they perform a function by the nuclear receptors. Their receptors are located on site of the nucleus to produce their action, or may be located in the cytoplasm. Agonist and antagonist. What are basic difference between agonist and antagonist? We can define as agonist. These are the drugs that occupy receptors and activate them. Basically, these are the drug metabolite. Okay, you can say these are the chemicals. Basically, they occupy the receptor sites and activate them to produce their action. What are the antagonists? Antagonists are basically the drugs that occupy receptors but do not activate them. Antagonists block receptors activation by agonist. Exactly, you can understand well that antagonists are also the drugs type, but they occupy receptor but do not activate them to produce their action. In the picture, you can see the agonist bind to the receptor and does the full activation of a receptor, and this produce their action. And there are certain agonist and antagonist. When agonist and antagonist they combinedly bind to the receptor, there is less functioning or less activation. When antagonists bind to the receptor, there is no action performed or no activation of a receptor takes place. What is affinity versus efficacy? Affinity describes strength of a drug binding with receptors. This is basically the capability of receptor to produce their action when a drug binding to it via the fit the lock. While efficacy describes ability of a drug bound receptor. This is basically describe the ability of drug bound receptor when drug bound to the receptor. What is the ability of a receptor to produce a response? This is called the efficacy. Turn the key. Okay, you can now understand fit the lock means affinity and turn the key means the efficacy. Agonists have both affinities for the receptor as well as efficacy, but antagonists have only affinity for the receptor and no zero efficacy, because you can say it fit to the lock but it doesn't produce its action or activation, thus it has no efficacy. All the agonists have no efficacy but also have tendency to perform or to lock with the receptor. Thanks for watching and kindly subscribe my channel that is by the name of Research is Alive.